Click the bell icon and subscribe to our channel so that you will not miss any classes. Hello students, welcome to the class. And now we shall continue with the types of cell division. The second type, that is mitosis. Here the term mitosis, the mitos refers to the thread and osis refers to state, which means that during mitotic division there is a formation of thread-like structures. So that is the meaning of mitosis. Here in mitotic cell division there is a formation of thread-like structures called spindle fibers. Spindle fibers are the thread-like structures which are formed during the mitosis, which helps in separation of chromosomes. So, since there is a formation of thread-like structures called spindle fibers, the type of division is referred as the mitosis. If you recall that in amitosis, there is no formation of such structures, thread-like structures. Hence, it is called amitosis. That means thread-like structures are absent. A means thread-like structures are absent. And the term mitosis was coined by Fleming. Fleming coined the term mitosis, and the mitotic cell division will occur especially in somatic cells. In multicellular organisms, especially which are reproducing sexually, they have two types of cells. One is body cells or the somatic cells, and the second type is the germinal cells or the germ cells. So generally the somatic cells will undergo mitotic cell division, which are having the ploidy 2 end. And in some organisms, especially in case of plants which are exhibiting alternation of generation, the somatic cells also may be of the type N, that is the ploidy. And in case of plants, as I mentioned, the germ cells also undergo mitotic division for multiplication purpose. That means to increase the number of cells, the germ cells of the plants also undergoes the mitotic cell division. Mitotic cell division is called equational division. It's very important. Mitotic cell division is called equational division. So you may ask with a question that why mitosis is called equational cell division? Why? Because during cell division, say for example it is a cell which is undergoing mitotic cell division and produces 2 daughter cells. Suppose if this parental cell is having 4 number of chromosomes, that means if, say consider it is a cell having 4 number of chromosomes. Once this cell undergoes mitotic cell division and produces 2 daughter cells, these 2 daughter cells also receives the same number of chromosomes. There is no difference in the number of chromosomes in the daughter cells which are produced. Hence, the mitotic cell division is also called equational cell division. So, you can give a reason that during mitotic cell division, the number of chromosomes in the parental cell and the daughter cells will remain the same. So, in 3 also the number of chromosomes are same. So, that's why it is called the equational cell division. And the mitotic cell division will takes place in 2 stages. there are two stages in the division. One is karyokinesis. Karyokinesis refers to division of nucleus. Division of nucleus, and it is a very dramatic stage. Division of nucleus is a very dramatic stage where the chromosomes will behave differently in different phases. Hence, the karyokinesis can be divided into 4 phases for the sake of understanding. They are prophase, metaphase, anaphase, and telophase. Even though we can differentiate karyokinesis into 4 phases, we cannot draw a difference line between the phases. That means these are the strict events which are taking place in prophase, which are the strict events which are taking place in metaphase, that we cannot draw. So that means the events in the phases may overlap, but for the sake of study, for the sake of understanding, they are classifying the karyokinesis into 4 phases as prophase, metaphase, anaphase, and the telophase. We shall take up them in detail further. And the second stage in the mitotic cell division is cytokinesis. And here the cytokinesis refers to division of cytoplasm. Division of cytoplasm is called cytokinesis. And there is a difference in the cytokinesis of plant cells and animal cells, and even there is a small differences in the karyokinesis of plant cell and the animal cell that we should take up at the end of mitosis. That means, as for now, I will explain only by taking the animal cell to explain all the phases, prophase, metaphase, anaphase, and the telophase. But at the end of mitosis, I will give you the difference between the mitosis of plant cells and the mitosis of animal cells. There is no large difference between them, but there are minor differences between the plant cell and animal cell mitosis that we shall take up once we complete with the phases of mitosis. And now, the first phase in the mitotic cell division is the prophase. So, to understand the events which are happening in the prophase, we must understand that before entering into division phase, the cell will just have completed the interphase, that is the preparatory phase. So, we shall draw first how the cell will be in the interphase so that it will be easy to understand how the components of the cell will behave during the prophase. So, I draw now the cell which is present in the interphase. This is a cell which is in interphase. Now, there is a nuclear membrane present. So, this is nuclear membrane. And in the nucleus there is presence of nucleolus, and the chromatin network will be present, that is chromosomes are present in a thread-like structure and we call it as chromatin network or just a chromatin, and this is nucleolus and this is cell membrane. And this is nuclear membrane. And there is presence of centrioles, and we know that the centrioles are divided in the interphase itself. So these are centrioles. So I am writing here only the things which are necessary for understanding the prophase. Of course, apart from these structures, there are many structures present in a cell. I am not focusing on that. Those structures which are necessary for prophase or the division, mitotic cell division, that only I am showing here. Now, once the cell completes the interphase, that is the preparatory phase, immediately the cell will enter into prophase. For the sake of understanding, they divide the prophase as early prophase and late prophase. Early prophase and the late prophase. So there are events which starts and they end. During the early phase, the events will start. By the end, the events will complete. So for understanding that, they divide the prophase as early prophase and the late prophase. So just now we shall understand what are the events in the early prophase. So this is the cell. There is no changes in the cell membrane, but the nuclear membrane will start disappearing. The nuclear membrane will start disappearing. You can see here there is a thickened and compact nuclear membrane present, but here and there the nuclear membrane will ruptures and it getting disappearing. So the first event is the nuclear membrane starts disappearing. Along with the nuclear membrane, the other cell organelles like endoplasmic reticulum, Golgi apparatus, nucleolus, they also disappear. They also starts disappearing in the early prophase. And the one major event which is taking place here is there is a condensation of the chromosomes. I told the DNA will be present in the network and we call it as a chromatin network. That chromatin network will starts condensating. Upon condensation, there is a appearance of chromosomes, and here the chromosomes were not clearly visible, but they just appear like a ball of wool. If you take a thread of wool and if you start winding it or if you start clumsing it, what it appears, just like that, the chromosomes will also start appearing in the early prophase. And I told there are centrioles which are already multiplied in the interphase will start producing a asters of spindle fibers like this. They produce the aster of spindle fibers. Aster of spindle fibers. And these spindle fibers are connected between the two centrioles. Some are continuous and some are discontinuous. That we will discuss or we will study in detail during the metaphase. But as for now, you should understand that the centrioles which are already divided during the interphase will start producing the aster of spindle fibers, and some of the spindle fibers will connect the two centrioles, and those connected centrioles will interact repelling action. They produces the repelling action. That means they repel with one another. As a result, due to the repelling action of spindle fibers, the centrioles will start centrioles starts moving to the opposite poles. They start moving towards the opposite poles. So these are the events during the early prophase. And here I have written only 4 chromosomes for the sake of understanding. To have a track of number of chromosomes, I am just taking 4 number of chromosomes. And don't think in all cells there is 4 number of chromosomes, and the number of chromosomes will differ from one animal or from one plant to the another plant. That you should have in mind. But for understanding purpose, I am just taking 4 number of chromosomes. And now after completing it, at the late stage, that means the events will gradually happen once the prophase reaches its end. Then what happens is, the events in the late prophase are the centrioles. They already moved towards the opposite poles like this. These are the 2 centrioles which are already moved to the opposite poles, and they have the spindle fibers connected with one another. And you should remember one thing. Here the first event is centrioles reached opposite poles, and here in this network we could observe the 3 things. One is the aster of spindle fibers and centriole, and along with the spindle fibers. Spindle fibers, some are connected with the chromosome and some are connected with 2 centrioles. So that I told that we should discuss later. And here this operators or this network, the operators consisting of aster, centriole and the spindle fibers is named as mitotic operators. And this mitotic operators is very important for the separation of chromosomes, that we should understand in the metaphase. And at the later stage, there is a complete appearance of chromosomes with 2 chromatids. So you should have a difference. See here, just the chromosomes are appearing like a ball of wool in the early stage. Once the prophase reaches its completion, at that time the chromosomes will appear completely by having 2 chromatids. This is one chromatid and this is another chromatid. Likewise, we are having we've taken 4 chromosomes, right? I am writing 4 chromosomes here. So this is another, and this is one more. 4 chromosomes are appeared. And the new cell organelles, that is nuclear membrane, nucleolus, endoplasmic reticulum, Golgi operators, all of them have disappeared. They disappeared already. So these are the events in the prophase. Once it happens, that is, once there is a completion of appearance of chromosomes, once the centrioles reach their end, once the cell organelles have disappeared, which is the beginning of metaphase and the end of prophase. That's why I told in the beginning, we cannot draw a strict line between the phases. Okay, so now in the next class we shall continue with the metaphase.